Start?
Yes.
Okay. Hello, everyone. Thank you for making it here on Saturday in the middle of the day. We're going to be talking about protein folding with AlphaFold and the 5 Nobel Prizes that were sort of all thanks to machine learning. Big W, as you can see. Okay. Uh-oh. There we go. So I'll just give you an overview of like what the Google slides are going to go through. So first I'm going to go through like an overview chronologically of the Nobel Prizes. And so the physics Nobel Prize was sort of about work done back in like 1980s with Hopfield networks. And I'll talk about John Hopfield, and later I'll talk about Geoffrey Hinton. And then that'll sort of lead us into the discussion about the chemistry Nobel Prize, which is a lot later, and it includes David Baker and the team at AlphaFold. And then I'll sort of talk about like what the actual problem is with protein folding. Like, why is it so hard to determine the shape of this protein, and why do we even care about the shape? And then I'll do a deep dive into AlphaFold 2, which is really like a step up change from all previous computational methods of determining proteins. And we'll dig into like why, if you're interested in the machine learning aspect, like what did they do exactly that made them so much more successful? And then if we have time, we'll discuss like what does this tell us about the future of AI, and like how can we use machine learning in science to do things like AlphaFold? Okay. Okay. So first, I'll talk about John Hopfield. So the main innovation that he was awarded for was Hopfield networks. And this is basically like a precursor to like our modern day neural networks. And he did a lot of work on like modeling the brain with math and computation. And so this paved the way for like an actual model of neurons and utilizing what the brain does in order to actually make a computational model. And so the brain is a machine, and like any machine, it can be understood through mathematics. I think that's a powerful line. Okay, and then feel free to stop me at any time and ask questions if you want.
What are energy-based models?
I'll talk about that with Geoffrey Hinton,
Okay.
because that's his main thing.
Okay.
With the Boltzmann machine, yeah. Yeah, and then yeah, Hopfield networks. That's what we'll discuss in combination with Geoffrey Hinton. So this guy is like the godfather of deep learning. He popularized backpropagation, and he was awarded the 2024 Nobel Prize in Physics for his work with Boltzmann machines. And also, just another side thing, he introduced dropout regularization, which is how it's a way to like prevent overfitting with neural networks. It's very important. He also won the 2018 Turing Award. And there's actually some controversy with the Nobel Prize because like they're awarding a physics prize to these computer scientists. But I'll sort of explain why I think it sort of deserves the physics prize, because it uses like the Boltzmann graph, and that is like a fundamental part of physics that is sort of proven with Boltzmann machines. So I'll talk about the energy-based model a little bit. So the network is structured to minimize an energy function. And so this is sort of modeled after like how actual molecules like go to different heat levels. And so if you look at this graph, like molecules will tend to spread out over different temperatures. And then over time, once they get to a certain temperature, like they evaporate away. And that's why puddles slowly evaporate. And so they evaporate and then go back up and then go back down and evaporate. And so that's just one example of like energy flow in the real world. And so he actually used this concept in Boltzmann machines to make stochastic neural networks. Does that sort of answer it? I know it's like a
Wait,
not
so
too specific.
are you saying that if you start out with all molecules being very similar speeds, they eventually spread out like that? So going from T3 to T1 is as time goes on.
Well, it could be, but I think this one is saying like T1, like the molecules are spread out, and then once the molecules like on the very edge, like evaporate,
Right.
they'll sort of flow back up here. So it can go both ways.
Okay.
But if there's nothing to like,
T1, T1.
if there's nothing to like change it to the gas phase, it will tend to just
Stay there.
stay there. Yeah.
Okay, I see.
Okay.
Oh,
Oh, yes.
sorry. So T is the temperature.
I think that's a good
Time.
question.
Oh,
Well, so the x-axis is temperature. It's the speed of molecules. And so
Oh,
the speed of molecules, if you're looking at it very closely, and that is what temperature is on a macroscopic level. And so I believe this could be either temperature or time, but temperature could be like,
I think it's technical because it's like T1 larger than T2 larger than T3. So it's basically like as temperature decreases, the sort of range of speed of molecules will kind of all go down to
Yeah.
this, and like you think of temperature was like as if you were to integrate, I guess, the
exactly,
curve
yeah.
because you're looking at the speed of all the molecules.
Yeah.
It's just the Boltzmann distribution. That's why it's called Boltzmann.
Yeah, it's the Boltzmann distribution.
The area under the curve should be the same, like this now, this is tracking the change.
Oh yeah, the arrow actually.
So it's trying to pick is like, as the temperature increases, like the average speed of the molecule increases, but also the spread is wider.
Yeah.
So like, if you say like, okay, like the boiling point is like, let's say like 10 degrees, like you have that as like the cut line, then the area under the curve for T1 is like wider than the first.
Yeah. And you're right about, so the area under the curve is sort of the number of molecules, because like the y-axis, the probability density function, shows like how many molecules are there at this moment or at this speed. And so, yeah, we'll have the same number of molecules, but they'll just be at different speeds. Okay. I don't want to go like too deep into this. We're going to mainly be talking about the chemistry Nobel Prize, which I'll get into now. So David Baker, I'll talk about. So he is sort of a pioneer for like earlier methods of finding proteins. If you guys are familiar with the Rosetta program, it was sort of like a way to get people involved in protein folding. If you weren't a scientist, you could just boot up this program and it'll try to chug along with these simple computational methods to sort of test out different protein structures and see if they work, see if they're correct. And so nowadays, David Baker was mainly awarded for his work in de novo protein design. So using our knowledge of how proteins are structured, he is trying to come up with proteins that don't exist naturally, to do crazy things like grow semiconductors. I don't even understand how that's possible with a protein, but like if we could do that, like that'll probably be pretty revolutionary. And so I stole these slides from David Baker's little presentation, but here's just an example of the potential of proteins. Like these are all natural proteins for any organism on Earth. And then here are like all the possibilities and combinations. But I mean, this doesn't even make sense because theoretically there's like an infinite number of proteins that are possible. And so theoretically, like we should be able to design proteins that do crazy things like grow semiconductors. That's what David Baker's work is on. And then I wanted to talk about the Fold It game, which David Baker also did as like an offshoot of the Rosetta program. And so it was sort of like anyone could log on to this and sort of try to do their best to improve the accuracy of a protein by like rotating joints. And it wasn't really too good because people didn't really know how to fold a complicated 3D structure. but I think it's important because I think it sparked something in Google DeepMind when they saw like you could gamify this process of folding a protein and getting it into the correct shape. And so they put down their board games and they're like, okay, we can actually solve this sort of game-like thing and actually do some really good science. Okay, so that brings me to AlphaFold, which is the main topic. And so John Jumper and Demis Hassabis were awarded the second half of the chemistry Nobel Prize. And here's just a picture of the team. Demis Hassabis is the CEO of DeepMind, and John Jumper is like the main head scientist for AlphaFold. And the reason I'm going to focus on AlphaFold is because it basically just solved protein folding altogether. So if we look at CASP, the CASP test, so the CASP is sort of, they come together every 2 years, and the committee has a bunch of proteins that haven't actually been released to the public, and a bunch of teams with computational methods of folding proteins try their best to take in this set of amino acid data and predict the protein. And I mean, before AlphaFold, like they're less than 40% confident that their shape is going to match up. And then after AlphaFold 2, they've reached like a 90% confidence threshold. And that is the main threshold where people are like, okay, this is the shape of the protein. Like you could basically use this. Yes.
How good are like trained biologists?
Okay, that's a good question. So, hello. Okay, so a trained biologist can use things like actual experimental data in order to like X-ray crystallography in order to actually get data on what the protein looks like. But that work is super expensive and it takes a long time. But people have like spent their entire PhD trying to like find the shape of one protein that's really important just through those like experimental methods. But like if we can solve it in a computer, like people don't have to go through that whole process. And now people can design drugs and do anything related to proteins just using this. Okay, so for anyone that's not like doesn't have a strong background in biology, I guess I'll just go through like what I mean when I say amino acid and DNA. I'll try not to bore you too much with biology, but the sequence of DNA describes how the chain of amino acids will look. And so there's things called codons on the DNA. It reads 3 letters at a time, and that'll define one amino acid. And long story short, if you know the DNA sequence, you know the amino acid sequence, and AlphaFold takes that amino acid sequence, and that's what it folds into the shape. And that shape is really important for knowing how it functions in the body. Oh, yes.
Did it focus on the secondary structure, the tertiary structure of the protein? Like,
Okay, so you're asking the secondary versus tertiary structure. So the secondary structure is sort of like alpha helices and beta sheets of a protein. And I guess the answer to that is it has to solve both of them. And so the tertiary structure is how the alpha sheets or the alpha helices and beta sheets come together in relation to each other. And those are sort of abstractions that scientists use to characterize the shape of a protein. But like a computational method is just looking at like this coordinate, there's an amino acid at this rotation. Yeah, so it doesn't really like structure it the same way we would think about it.
So what's like the input out? So is the input just like a sequence of like A, T, G, and C, if I remember correctly. And the output is just for each of those A, T, G, and C's, where is it in 3D space?
Yeah, where's the corresponding amino acid? Yeah.
I see. Okay.
And I'll sort of explain why shape is so important. Some of you may have seen this diagram of an enzyme. So basically, like chemical reactions are how our body functions, and proteins are how our body like undergoes chemical reactions. So if a protein is the wrong shape, the chemical reaction wouldn't actually be able to happen. Like the enzyme doesn't just help the reaction happen, like it is necessary oftentimes. And so that's why understanding the shape, we'll be able to understand the chemical functions in our body. And then that sort of leads us into the protein folding problem. So why is it so hard to like fold a string into a little blob? Well, I think a big reason why traditional computational methods were so bad at this is because if we look at like the ionic bonds and the different types of interactions among amino acids, if any one of those is slightly off, the angle will be slightly off, and the next one will be slightly off, and so your errors really compound, and you're not able to get the actual shape.
Wait, so is a single amino acid is either A, T, C, or G?
Oh no, sorry. So A, T, C, and G
Yeah,
are nucleotides on DNA.
okay.
So they're like they code the amino acids,
I
but
see.
there's 20 amino acids. So there's 20 amino acids in humans and in most life on Earth. And we can represent those by letters as well,
Yeah,
like glycine, and so on.
yeah, but each of those has a unique.
It's like this. It's on the table.
Oh, okay.
This chart.
Oh, I see. Okay.
I'll go back here. Yeah. So the DNA has just 4 letters. And then so there's something called the mRNA, which reads the DNA, and then the mRNA transfers it to a ribosome, which will actually translate that. The T's are turned into U's for some reason. And then the amino acid will be added. So it always starts with a methionine amino acid, and then it'll read until it goes to a stop codon, and then it'll detach from the ribosome.
Right. So okay. So basically, you have the DNA with the code tells your body what amino acid to put in what order.
Yes.
Okay. And
In
you're
general,
basically
yeah.
trying to say, given this essentially coding of what your body is going to do, can you predict what it will end up doing?
Yes,
I
exactly.
think.
Yeah.
Okay.
Okay.
I see what biology.
Okay. So that's sort of like the general biology behind the problem. So now I'm just gonna go back to like what was the first instance where they found the shape of a protein? And it was myoglobin, which is a pretty interesting protein because it's just one sequence. And usually things like hemoglobin, there's like 2 different sequences that come together to form hemoglobin. But myoglobin is just one sequence, and through a lot of effort and using X-ray crystallography, they were able to find the shape of it back in 1962. And these 2 guys, Kendrew and Perutz, were awarded the Nobel Prize for finding one protein shape. And that was a lot of work. But if you guys are willing, I want to take us through a live demo using AlphaFold. And I want to show you how easy it is to find myoglobin now that we have this computational method. So I'm gonna do it on my laptop, but if you want, go to the tiny URL and you can copy paste the FASTA sequence, which is this sequence is all the amino acids. So if we go to AlphaFold, copy paste the sequence of amino acids, like it will hopefully spit out the shape that took these guys so much work with X-ray crystallography and everything. Okay. So if you guys are able, that link will take you to the slideshow. Go down to slide 17. And so you might have to log into the AlphaFold website. And then you should get somewhere over here where it says protein, 1 copy, and then paste in the FASTA. And so you're going to go and copy paste this sequence. And hopefully it'll take a while. We'll just say, I'll just name it myoglobin. Okay. Now the job has been launched. Okay. So right now AlphaFold is working on it. AlphaFold has a set of weights already. So the machine learning's already done, but now it's just using that algorithm to take in our sequence and try to make a 3D model. And so I'll pull up what the model looks like. Oh, that's in the way. Okay. So this is on the protein database. This is what we know myoglobin looks like experimentally. And it's going to be sort of hard to tell.
What's the big parts and the thin parts?
Okay, yeah. So I'll just explain this right now. So this is myoglobin. These are alpha helices. And I believe this, yeah. So, and then the thin parts are just strands of amino acids. And the alpha helix is like a strand on a strand on a strand, like looping back and forth to make a sort of sheet-like thing. And I believe this is where a heme would be. And a heme is like a metal that is inserted like somewhere on the protein, and it carries some like oxidized element around the body. But okay, let's see if our AlphaFold thing is done. Okay, yes. So we just pasted in that sequence of a bunch of letters, and AlphaFold gives us a 3D structure. And if you'll notice, it's kind of hard to tell, but these are like the same structure. Like it just gave us the protein that took these guys a bunch of effort, and they won a Nobel Prize for finding it. But we just found it with AlphaFold too. So I just wanted to demonstrate like how much of an innovation this is to have a computational way to do this. And like before AlphaFold, like I said, people were spending entire PhDs to find the shape of one protein. So I don't think people realize like how much of an innovation it is to be able to just input this sequence and get a 3D structure. And so, yeah, if you can sort of see, there's one end here and one end here, and we'll try to line it up with what AlphaFold gave us. I think that's what it looks like. Yeah, one end here, then you have like the strands. Yeah. And so I'll just talk about like what else is AlphaFold trying to tell us here? So the yellow parts means it's not actually that certain about the structure. It could be that AlphaFold like got it wrong and it's not certain, or it could just be that there is no defined structure. Like it could just be a loose, floppy bit of the protein. And then this is a whole map that you don't have to fully understand, but it's just about the expected error in angstroms. So like on the atomic scale, how far off might it be? But okay. So that was just a little demo of using AlphaFold. I'll get back to the presentation.
Protein, like the actual protein is that there's like a heat that like sort of like stabilizes the protein.
Oh, yeah.
Doesn't have it on the amino acids.
Oh yeah, that's a really great question. I was gonna get to that, but I could just explain it now. So he pointed out there's a heme here on myoglobin that like isn't included in the amino acid sequence. How does AlphaFold know how to shape the protein if it's affected by this thing that we didn't even specify? And the answer to that is AlphaFold isn't actually using like physical repulsions in order to predict the protein. It's using a lot of evolutionary data from other species in order to tell which amino acids are likely located close to one another. And I'll explain that later. That's a big concept that's sort of complicated, but
But that gene is present in all amino acids and across all animals, and like those proteins are gonna be that way.
exactly, yes. So like if there was a mutation in this amino acid and this amino acid, they're close to each other physically, but they're really far apart in the chain. We can sort of tell AlphaFold knows, oh, these are probably close to each other if they're both being mutated together across different species. I'll explain that more clearly later on. Yes.
So this is like, it's exactly folded into this specific shape, like no deviations allowed in the evil or.
So that's a good question. So can there be other ways that the protein is oriented? The answer is yes, but oftentimes, I mean, I don't actually know. I can't say too certainly. Like you should probably ask one of the Nobel laureates about this, but I think these angles and like this shape is sort of how you would see it if you had like a vat of this protein, like, or just how it exists in this cell. But if you raise the temperature, for example, it'll denature. If you raise the acidity, it'll denature and the shape will change. So there's other things to take into account, like the kinetics of it, like how it will change in different circumstances. And that's sort of what like AlphaFold 3, the next version, is trying to do. Is like how does it dynamically change? Because you could probably tell that's important in chemistry because things are dynamic.
Or maybe this is just at like some kind of base temperature.
Yes, yeah. So like at maybe the specific conditions that we set, room temperature or whatever, this is what it should look like. Okay. So now I want to dive into the paper. So the main paper on AlphaFold 2 is just called Highly Accurate Protein Structure Prediction with AlphaFold. And if you want to follow along, I have the link. You could go on the slides or scan the QR code. You know it works. Yeah, I've already showed you guys the slides, so hopefully you're able to navigate there. But yeah, so highly accurate protein structure prediction with AlphaFold. So I'll just go over, first of all, AlphaFold is really, really good. Like, if we look at this thing, like there's hundreds and hundreds of amino acids, and like any deviation in angle earlier on, like is accounted for and corrected for in order to get things that sort of resemble each other at any place in the entire molecule. And this is like, this was mind-blowing when they first released this because it's sort of like all the previous computational methods weren't able to do this. Like they would fall apart. Uh-oh. And here's just a big percentage that I wanted to put up. From 2021 to 2025, there was an increase in 118,900% in the known protein structures. I mean, yeah, that number is just astronomical. It's like a huge step change, and it changed like the entire paradigm of how we know about proteins. And so oh yes, questions.
That like figure based like experimentally verified like genetics, or is it just predicting structures that haven't been tested?
Yeah, so like we haven't added AlphaFold predicted structures into the PDB, the protein data bank. So we haven't actually increased by this astronomical number in experimentally proven proteins, but people can still use all these proteins that we've found with like 95% confidence, and it'll even show you like where it's confident, where it's not confident. So these are proteins that are actually being used. Did someone else have another question? Okay. But yeah, and then people are working to take these predictions and prove them experimentally and add them to the protein data bank. And so here's the graphic again of CASP14. And I also added the CASP scores. So CASP, once again, is the sort of conference where they meet once every 2 years. And this is AlphaFold 2, and this was second place. And so, I mean, they really just blew the competition out of the water. Every single previous year, they blew out of the water. And at a certain confidence level, you can sort of just say, like, this is the protein. All right, now don't get scared. This is the image that you see once you first open up the paper. You don't have to understand all this. There's a lot of arrows and words that I don't fully understand, but I'll sort of go through what in general this is. So at the end of the day, we have the input sequence. So the human DNA, or even just the sequence of amino acids. And by the end, we have the 3D structure, as well as like how confident it is. And so I'll sort of dig into, I won't go too far into the nitty gritty of how this whole structure works. But I'll explain what is MSA, multiple structure alignment. What are we pairing here? And I'll talk about the evoformer, which is sort of like a transformer. It uses attention, but it's not exactly a transformer, and I'll explain what I mean. Yeah, evoformer. Oh, I skipped a slide. Okay. So basically, this sums up what the last image was. If you don't want to understand all of the machine learning behind it, you could just understand this. So we have the input sequence. We have the physical and geometrical properties, how amino acids should line up. And we use evolutionary history in order to better our understanding of how the protein looks. And so, you have a question? Okay. So evolutionary history is the MSA. So the MSA aligns the amino acids of a bunch of different species. It lists out the human and a fish and a rabbit and a chicken. And we know that these sequences function and they work in these animals because they're alive. But like there's parts where the amino acids aren't the same. And so we know if we look at where the amino acids aren't the same, we see that they're pairwise. Like if this one changes, the other one changes at this location way down the road. And the reason that is because if amino acids are interacting and they need to have a strong connection and they're forming an ionic bond, let's say, well, if one of them gets mutated and the other one stays the same, the whole structure will fall apart. But if they mutate together, they'll still have the connection and the animal will still be able to live. And yeah, so that's how we're able to determine, like, through evolution, which amino acids are probably next to one another.
You can use the ones of like in the humans and the fish that are like really now.
Yeah, I mean, yeah, even humans will have mutations. Like, not all humans have the same amino acid sequence. So there's a lot to use there with these differences in amino acids, with the knowledge that these proteins still function in the same way. Did you have a question?
What's it mean by like the database search? Does it like find similar like
Yeah.
proteins?
So there is a database of the amino acid sequence of a bunch of animals. And so AlphaFold just takes in that sequence information and compares it to the human amino acid sequence of interest. And or this could even be, let's say the myoglobin sequence that we wanted. Or just some sequence. And it compares it on a array to all these different species. Yeah, I guess I already explained it, but yeah.
Is this a big picture diagram for the training process, the inference process, or like?
I'm not too familiar with like the exact difference between inference and training,
'Cause,
but
like, there might be like more information you can add like during training, for example, but like might not necessarily, because like, would you also need to do the genetic database search to look for similar sequences during inference, or would it come through with the separation?
that's a good question. I believe this is all in training, and then I don't quote me on that, but I think this is in training. It goes through this in order to tune the weights of the model. And then in inference, when we paste in our sequence of amino acids, it uses those weighted neurons in order to find a 3D structure. Yes.
It seems like the different species are part of the input to the evil format level.
Yeah, you're talking about this arrow.
Or like into the actual model with the evil format in the top half, like MSA representation.
Over here.
Yeah, so that would need to come from the database, right?
So I'm gonna talk about sort of this MSA representation where we're now recycling like stuff from the pair representation. And after the Evo former attention blocks, like there's more that gets recycled in order to adjust the MSA. But in terms of like what's input, I would say it's from the known data bank of amino acid. Yeah.
I don't know. But I don't know, it seems to me that those like lines, the solid lines are used both for training inputs and maybe those like, like sky lines or for training like this. Does it relate to that or something, or is that why the arrows are close down? Not entirely sure. So
That's a good point. Yeah. That could actually be it. I would have to read more into the paper, but that's actually a really fundamental question. Like, are the blue lines taken through in train? We know they're taken in through training, but then at inference, does the sequence go through this whole process? I'll have to look at that. Yeah. Okay. But that is like the big complicated picture that I didn't want to like dive too deep into. But okay. So now I sort of already explained MSA representations, but so it uses evolution to detect which amino acids are likely close to one another. So if a bunch of different species have an R amino acid, but then they also have a D amino acid on this other part in the sequence, it's really interesting that these two changed in unison, and that tells us that they're probably close to one another. And I say probably because like the MSA representation isn't definitive. It's still saying like there's a probability that these are close to one another. And so if you're interested in the math of the MSA, this basically just tells you like how we're defining it, because like an amino acid sequence on one animal might be a little longer than the amino acid sequence on another one. So how are we actually aligning them? And so this basically describes how we create gaps in order to actually make sure we're talking about the amino acids of interest. But I won't dive too deep into that. Okay, now I'll talk about the pair representations, which are over here. So the pair representation is sort of its guess on the spatial dimension and the geometry of the amino acids. And so this is sort of how AlphaFold grounds itself in Euclidean geometry. So like if I know amino acid 1 is 2 units away from amino acid 2, and amino acid 2 is 3 units away from amino acid 3, then I know that 1 and 3 have to be somewhere in between. It can't be like 7 units away for amino acids 1 and 3. So I mean, it grounds it in like the triangle principle. Yeah. Do you guys have any questions on the pair representations? Yeah.
What actually goes into those like cells in that pair representation? Is it like the distance made strokes? Like each of them has-
Yeah. So it's the
But isn't that what we want to find out?
so it's okay. So the pair representation starts out at a singularity. So in the beginning, I forgot what the name was. It's like singularity. I forgot the name, but they start out at 0, and after like looking at some evolution data, it will just expand out a little. And so, but the pair representation will always be like consistent with geometry. But the evolutionary data is sort of not really grounded in any geometry. It's just a probability of which ones are likely next to each other. And so that's why when we combine this set like information on distances with evolutionary probabilities, we're able to get a 3D structure.
I guess my question was more so that like if the 3D structure is dictated by how far each of those amino acids is from each other. So presumably that should be the output of our model.
I think it's like the initial guess.
Yeah, it's the
Oh.
initial guess. Yeah.
And like I think when I saw like AlphaFold like a long time ago, they have like it like predicts iteratively. It was like many iterations.
Yeah, exactly.
Oh.
I should have put.
It's
I
like recycling, like the thing where like it just like recycles the current output and then puts it back in until like it gets a better result.
Yeah.
Or something.
Yeah, I think I actually read that it was. Okay, I thought it was 8 rounds of recycling, but it might be 3 rounds. But it will guess the protein structure, and it's very wrong on its first go because it just set all of the values for the pairing to 0. But then I should have put a graphic up, but it'll basically like balloon out and sort of define the shape.
Okay, so
Going
each
up.
number in that grid is going to change with each iteration as it gets like more as it gets
More
better.
accurate. Yeah. And then each successive time it predicts a structure, it'll use that, make a pair representation, and use that newly informed information to do the next round. Oh yeah, this sort of explains it. So yeah, we have the 3D structure, and there's an algorithm to take that 3D structure and turn it into this pair sequence. Okay, and so now this, I'm not gonna lie, I really don't understand like the Evo former too well, like the row wise and column wise attention, but it is a sort of attention like framework, like a transformer that takes in the MSA evolutionary data and the pair representation that we've guessed, and through attention sort of relates these two in order to get a better informed outcome of the MSA and pair representation. Yeah. Do you have a question? Oh, okay. And so if you're more familiar with transformers, you'll probably understand it better than I would, but there's 48 blocks, so 48 neural nets that keep on feeding into each other. And by the end, the MSA that we get out will be sort of more informed based off of the geometric properties that we have in the pair representation. And the pair representation will be more informed on the evolutionarily which amino acids are probably close to one another.
So with because we don't know the pair representation of a molecule, or so that we don't know the pair representation of an inputs because that's what we're trying to predict.
Yes, exactly.
So what do we initialize it? That's what we're actually trying to predict.
So that's like the singularity initialization. So they just initialize it to 0 to
Okay.
start off with.
See, everything is just
Yeah, everything's a singularity. The protein just looks like a point.
Yeah.
And then later on, once we go through the whole process, it'll expand out a little bit and it'll still be really incorrect.
yeah.
But over time, it'll get better and better.
It's supposed to be confusion.
Mm.
That's where the stiff connection is because if the pair representation is all 0 at first, the result of the process is just going to be the same as the involved distribution. But then over time, as it becomes like more and more non-zero, it's
What is this one?
It's
Mhm.
supposed to be about four. That's also
Okay. So then there's also something the paper mentions called triangle self-attention, which is for the geometric pair representation. And it sort of looks at if amino acid 1 and 2 are this distance, and amino acid 2 and 3 are this distance away, we know something about the distance between 1 and 3, and so it sort of attends to that. And it sort of does that process on a bunch of different amino acids. And that's sort of how it goes from this singularity pair representation into a better informed thing that actually makes sense for a protein. And they also have this, which I think is really a really important takeaway if we want to make things like AlphaFold 2 in the future. And it's what this chart is, I believe, is sort of how successful the model was with different techniques for machine learning. So with self-distillation training, we can see the GDT difference was actually really good. And the IDDT, which is forget what exactly it is, but it's basically how good the model was at predicting the structure, was also really good. And so it goes through the different methods that the neural network uses and how good is it at each one of these. And so no IPA, it's really bad. We've introduced that, it gets a little better, and it gets better and better with all of these methods that they've used. And so I think if you want to like do your own research and develop something like AlphaFold, this is a really key takeaway.
what is like NPA?
So I believe IPA is a way to like filter in machine learning. I'm not quite sure. I don't know the intricacies of all these different like machine learning techniques, but if you want to go through the paper and learn them, they're probably really useful if you want to do something like AlphaFold. Okay. And so there's one last takeaway that they make about the intricacies of the MSA representation. So they found that when we have amino acid sequences that have more like connections with each other, like there's more times when an amino acid changes here and changes here, and we could detect that difference. Like how good the structure was actually increases. It's not too strong of a correlation, but you need a certain amount of what they call median per residue and for the chain. Multiple sequence alignment. So this is the evolutionary table that it's basically this or this. So the MSA Yeah,
Right.
It's
it's
actually derived from the database. It's not just like an embedding, but it's actually from the database.
it's from, yes. So we can get the MSA straight from a database of all these different species amino acids.
Oh,
Yeah,
go back one slide to the one with bold and, yeah.
this one.
So we had to look at two different databases for, and they're not derived from each other,
Right.
like the structured database and the genetic database, they're not. Are they like separate things or like is one?
Oh, so I guess I didn't really touch on this. The structured database isn't actually used. It might actually be used to initialize if we don't want to start at like a point singularity, but it gives us a template for the pairing. So
It mentioned templates in one of the steps in like what when the graph, or not really graph, but the diagram that shows like each step that makes the model better, it showed templates. But it doesn't look like, or templates might show up in the step here, but I don't know.
yeah, that's a good question. So yeah, I mean there is I guess they do use a database of other protein structures in order to get a sort of well-informed pair representation.
So that's like the initial step. So they have the option to not choose Starbucks.
That's a good question. From what I could tell, they use a singularity. But I might want to look into that. Like
And that's AlphaFold 1. Or is this 2?
this is AlphaFold 2.
Oh, this is 2, okay.
Yeah, this is the really successful AlphaFold model.
There's a plus there, so I guess if there's no template, it's just like the singularity where they define everything.
And AlphaFold 1 was able to sort of make some sort of structure. And so, I mean, if we look back at the AlphaFold 1 paper, I'm sure there's a whole other set of techniques and mechanisms they use to sort of make a pair representation that's sort of good. And once we have that, we use it and attend to the MSA in order to get a really well-defined structure. But I mean, yeah, there's so much here that I don't even fully comprehend it, and so that's why I encourage you to go read the paper and try to understand all these intricacies. And this is the Evo former. Oh yes.
And you know what they use for the loss function? It's just like the global position, or is it like the relative position to like the previous?
So that's a good question. Like, what do they use as a loss function? So I know for CASP, they have a really well-defined, like, how far off is your structure? Because they take in, this is the position of the amino acid, this is where you said it should be, and this is where it actually ends up being. And you're off by this many angstroms. And it sums up like you're off by this many angstroms on this one and this one and this one. And I'm sure there's some algorithm, some common algorithm they use to see what is your error. Yeah.
Is that on the 3D structure or on the very final pair representation? The pair representation
Oh, yeah.
gives you-
So, I mean, yeah, here's like the score residue, which is sort of resembling of the pair representation, but it actually shows you a score. So I guess like how well it was predicting. And I think it uses, I mean, this is sort of like a confidence measure. It's because it doesn't know what exactly it should be, but this is like a confidence measure at a certain point. But like with CASP, they actually had they knew the structure, and so they're able to determine from that how far off you were. But it's a really good question. Like, how do they know that they're not really confident on that end, but they're really confident on this little swirl? Yeah.
So CASP is just their like annual contest, right? Like even prior to AlphaFold.
Yeah. So
Like that.
yeah, so protein folding is like a 50-year problem, and CASP happens every 2 years. So they've been doing this for 28 years before AlphaFold. and they, yeah, so basically they would just have a secret set of proteins that they found the structures for, and then have the computers take in a sequence and try to predict the structure. And they were pretty bad up until AlphaFold. But this, since we don't actually know what the structure looks like, we're only able to get a confidence measure, right? Because if we knew what it was, we would just show that. Okay. Are there any other questions? I know it's very confusing.
What's the main improvement between AlphaFold and AlphaFold 2? Like
AlphaFold 1 and 2.
yeah. Is AlphaFold 1 also like a transformer situation, or they
No,
it was just
I don't believe so. Well, yeah. So AlphaFold 2, the big breakthrough that they advertise is their Evo former, and so they say they've invented this new transformer-like model with 48 blocks that takes in these two sets of data and gives us a 3D structure at the end of the day. But yeah, I should actually look more into AlphaFold 1 and like what specifically they improved.
You know how they turn the pair stuff into the structure?
The pair representation into the structure. It uses it will, so the structure module is a complicated algorithm that uses both of these in order to get its best guess at the structure.
It's like another attention. It's pretty interesting.
I have to look back at the paper. I don't know. It could be yeah. But I think their main innovation was the Evo former that made the pair representation usable and resemblant of the pair representation we should get from the 3D structure. Yeah, you have a question?
So is it sort of like two towers and you're transmitting information between each other between the system? That's what the is like.
So you have these two towers. I wouldn't say so. You're not attending between these two. You're attending between the pair representation and the MSA representation.
Two representations. One model focuses on pair representations. The other maybe like focuses on the structure. And then you're sort of like transmitting information.
Yeah.
Similarities.
Yeah. So at this red amino acid, we're attending to like the red block over there that corresponds to it, and seeing how do we mold it to be more accurate. And so they're attending to each other, and I think, like we said, it attends to the MSA first. Yeah. So I mean, this is the Evo former. If we like expand it out more, there's row-wise gated self-attention with pair bias on the MSA representation. So it takes in the pair representation and attends to the MSA in order to get something that looks better and better, in order to finally get a better MSA representation. And then after we have a better MSA, we take that and attend to the pair representation, and use the principles of geometry to get a better pair representation. Yeah.
Why would we need anything other than a pair representation, final pair representation, to generate the 3D structure? Because if you know how far each amino acid is from every other one, then that should be enough to generate the final structure.
So that's a good question. Like, why do we need the MSA as well to determine the final structure? Like, doesn't the pair representation
like at the very
tell
end?
us? Yeah. I think the answer is that the pair representation is still not, it doesn't fully describe the structure because it is still a probabilistic model of distances. And this is also a probabilistic model of which amino acids are close to one another, and etc. And so at the end with the structure block, you still need to combine these two in order to get your best guess, is how I understand it. Yeah. Okay. So we could probably talk more on the Evo former, but I've honestly like reached the limit with you guys like on how much I fully understand about it, because there's so many intricate parts that I don't actually fully know. So now we could just sort of discuss like what's the future of protein folding. So we have this static 3D image of the structure, but that's not really all that a protein is. And so Demis Hassabis, who's the CEO of DeepMind, which runs AlphaFold, says that they'll be using AI to develop drugs. And he also really wants to simulate a virtual cell. That's sort of been his like long-time goal, and that's why he wanted to make AlphaFold. Because if we're able to fully simulate every single aspect in a human cell, like, can you imagine the implications that would have on testing a drug, for example? Like, we just develop it on a computer, put it into this virtual cell, and see what happens. We don't have to go through a clinical trial with human patients and see what it does. We just have it on the computer. And another thing is with David Baker's de novo protein design, like there are so many proteins that haven't been created naturally, and there's a really good chance we could use them to do things like solve climate change or cure cancer or create semiconductors somehow. And also just everything to do with biology is probably going to be improved by this knowledge of proteins, like CRISPR, for example. If we know how a protein looks like given a DNA sequence, this will probably help us out with CRISPR, which edits the DNA sequence in order to give us a protein that we want. Do you guys have any thoughts on the protein folding problem? Any questions? Okay.
There's like a new thing which is like RNA folding. So it's like another thing they're trying to fold.
RNA folding.
I saw a competition on Twitter.
Oh. Yeah, I mean, so RNA is sort of broad because there's messenger mRNA, which is still made up of nucleotides, but then there's also ribosomes, which are technically RNA, but they're also just big proteins that make proteins. Yeah, do you have a question?
Also, can you go back to the slide where they described this genetic section and there's at the end? So they put that.
Wait,
so,
this one? Or
yeah, no, the one. So why did they use like, I guess like genetic data instead of like amino acid sequence as itself? Because I think like for the third representation, it's like it describes like tertiary slash quaternary, right? Because like it's described after it has fully folded. I believe it's tertiary because I think for now we're looking into a single amino acid sequence, but why don't they just use like just the amino acid sequence? Like why are they using like genetic sequence? Because even if like amino acids may differ, I mean, sorry, nucleotides may differ, like like there are like codons that code the same amino acid. So wouldn't that be more like happening?
well, the MSA, remember, this isn't the DNA. These are amino acids, right?
But is this genetic data? Are those amino acids?
Yes, these are amino acids.
Oh,
So it takes amino acids from these different animals. So these are different sequences of proteins. Like a fish has a different sequence of proteins in this example than a human, but the proteins still do the same function in their body. So we're able to use that information in the MSA. Did you have a question?
Oh no, I was gonna talk about future, but
Okay, yeah, we can talk about that.
that's actually really cool that there's RNA folding in animals because like you're saying for ribosomes, what we're finding now is that a lot of ribosomes, like RNA, especially, it's not just like messenger RNA, like you're saying. There's also tRNAs that are actually like methylation RNAs that like are used in the ribosome to like transfer amino acids to like attach amino acids in the polypeptide. But then not even just that, there are also ribosomes that we found that actually catalyze reactions. So for example, like the ribosome, the key reaction in a ribosome is the transpeptidation, where you're moving an amino acid. Well, you're moving the amino acid chain that you're building to the new amino acid, like tRNA, and that's actually catalyzed by RNA. So the fact that there's also reactions that are catalyzed by RNA as well, not just proteins. That means there's also interactions. The cool thing about RNA ribosomes as well is that there are proteins in it too, but they either catalyze different reactions or they're structurally, well, they're there for structural integrity. So then there's also interactions between RNA and proteins. So not only are you dealing with binary structures where you're binding different proteins together, you're also binding different RNAs with proteins or RNAs with RNAs.
Yeah. Yeah. So proteins are a huge part of biology, but it's not the whole story just to know what the shape is. And so that's what a lot of this research is going to look like going forward. Like there's other parts of the cell that we need to understand. And also like the protein changes over time and in different circumstances, it'll fold a different way. And that's also another thing that we have to understand. One cool example that I remember is people were actually able to hack AlphaFold 2 in order to get like complexes that were 2 protein chains. So like hemoglobin, for example, like it combines a beta and an alpha in order to make the whole structure. But people just put the sequence for the alpha, put a bunch of glycines that are floppy and don't really form much structure, and then put the next sequence. And AlphaFold made hemoglobin by just combining these 2 sequences with a floppy glycine chain in between. So I thought that was interesting. Yes.
I would imagine that like for simulating a virtual cell, like get the added complexity that different like proteins can like enhance or inhibit like the creation of other proteins.
Yeah.
If like they affect how they each get like created in the first place, and like if environmental factors that like change the shape of mRNA and stuff, like that would affect how the transcription and translation happens.
Yeah.
Like, I don't know. It feels ambitious to go from like now we know how to create like individual proteins to being like now we can like simulate how like millions of them.
That's true. Yeah. I mean, I still think we're pretty far away from simulating a virtual cell because there's a lot more computation, I would think, because every atom is interacting. And I mean, that's just really hard to compute. Do you have a question?
And also like this research, but I think they're just focusing on like making like tertiary structures, so like they include like post-translational modification.
Yeah, exactly. Yeah. Like in some proteins, like their amino acids get cleaved off in reality, and the final structure is something completely different than how it was synthesized. But one thing I want to mention is the DNA sequence has spaces in it that are called introns that aren't actually translated to become the protein. So there's a whole methodology to determine like which DNA codons are actually going to become amino acids. And I think a lot of that also considers after synthesis, like the methionine that first gets made is oftentimes just cleaved off. So we can just consider everything after the methionine in the amino acid sequence. But in general, yeah, things are so dynamic that it's hard to tell. Okay. And now we can sort of discuss like, are there other applications for machine learning besides protein folding? I think the answer is obviously yes. What do you guys think? Do you think attention is here to stay? I mean, the Evo former was clearly really successful, and it utilizes something that's very similar to a transformer. So I don't know. It looks like attention is here to stay. Yes.
I'm not very familiar with this, but from the papers I have read from their class, I remember for some DNA stuff, people have been moving on to like Mamba and Stripe Hyena, those kinds of things,
Mm,
right?
Mamba.
Could that be applicable towards protein folding, or is that like a not very related?
You'll have to remind me what Mamba is.
It's state space model. So it's basically, it's linear. Except for the gravity.
Like fancy RNNs.
Yeah.
Ooh. Yeah, I don't know. I'd have to look into that.
It's already like that long, right? So I don't know if that's cats.
A little bit, they're like extremely long.
Oh.
They can be. They can be. I mean, it depends on. I mean, it's not like all of the information that gets dumped into an LLM type of long, but it's certainly hard to compute all the combinatorial ways that the protein could look. Like that is enormous. Another thing I want to mention is, I mean, there's other areas where attention is looking promising, like EEG signal processing, for example. And I'm sure there's other examples where machine learning and attention are going to like revolutionize the field. I don't know if people have recognized. But what do you guys think? Do you think there's going to be AI agents that are able to do research on their own? I don't know. Are we going to have a job? Yeah, I don't know. Like, is AI? I mean, AI is a lot of different things. Is it? Like, yeah, I don't know. If we're
Oh no.
I think it could go in a lot of different directions. Yeah. But yeah, so yeah, a lot of people think it's.
But shouldn't be in the apocalypse and paradigm like intersection.
I put it in an apocalypse because like robots are better than humans now. It's like a landmark moment. I don't know. Yeah, like how would you guys order this Venn diagram? It's interesting. But yeah, I tried to tell the story of AlphaFold just because I think it's one of the best examples of machine learning that is revolutionizing science, and it's not just slop or gonna ruin the world or anything. Yeah. Okay. And that was the end of my slides. Yeah.